It's my pleasure to invite the second speaker, Dr. Anil Kumar, who is a staff scientist level 5, and he is in Gene Regulation Laboratory, National Institute of Immunology. And he is going to speak on towards investigating the anti-cancer role of gut microbiota-derived metabolites. He has published more than 75 in publications in international journals, and he has participated in the development of biosensor, which has now been transferred to the industry. And he also has one of his patents selected for CSIR technology. He's heading the IPR division of the National Institute of Immunology, and he has been granted more than 50 patents. So the stage is all yours, Dr. Thank you.
Thanks for kind introduction. Thanks to Professor Ganguly and Dr. Nirja for inviting me here for this event. So today I am discussing about implication of gut microbial metabolites in the causation and treatment of cancer. Particularly, I will focus on colorectal cancer. So as you all know, we are not alone. We have a lot of bacteria, a lot of fungi in our gut or on our body. And you will be surprised to note that more than 10% metabolites which are flowing in our blood are actually coming from different bacteria, different fungi, short chain fatty acid, as we were discussing. So, and other metabolites like TMO, TMA, all these metabolites are coming from bacterial and fungi origin. So, these metabolites are playing important role in human health and diseases. So today we know what kind of bacteria we have, what kind of phyla we have in our gut, but we don't know much about metabolite and functions of metabolites. So last 10 years have been devoted to identify what kind of metabolites are there from different bacteria and fungi. So with the help of mass spectrometry and NMR, we identified a number of metabolites. And today we know more than 200 metabolites, which are quite important. And these metabolites like short chain fatty acids, trimethylamine oxide, trimethylamine oxide, paracrysyl sulfate, indoxyl sulfate, phenyl sulfate, all these metabolites are playing very important role in making us healthy or causing different diseases. If these metabolites are not in physiological range, then there are implications in different diseases of these metabolites. So these metabolites, when they are produced in the gut, they may have a local effect. They can interact with the intestinal cells. They are responsible in maintaining gut barrier function. They can, even if metabolite like colibactin, they can damage DNA. So, and these metabolites may also have a direct systemic effect. They can go to heart, brain, lung, and liver. Okay. So if they go to kidney, metabolites like EPS or PCS, indoxyl sulfate, these are uremic toxins. And these metabolites can interact with albumin, and they can cause chronic kidney disease or other kind of kidney ailments. Many metabolites go to liver for conversion, like trimethylamine. Trimethylamine get converted to trimethylamine oxide, and then they have their physiological functions in human body. So these metabolites have roles in human health and diseases. These metabolites are also important in our immune system, like indole derivatives or short chain fatty acids. These metabolites interact with the different immune cells. These metabolites are responsible for maintaining tight junctions of gut lining, and they are also important in T-reg differentiation or T cell activation. They interact with different kind of immune cells. So these metabolites are quite important in maintaining our immune system. So my talk is related the link between cancer and microbiota. So cancer has different different causation factor, but one factor is biological carcinogen. So according to WHO, more than 15% of cancer diagnosed globally are attributed to biological carcinogens like bacteria and viruses. We all know about Helicobacter pylori, which cause gastric cancer, and Epstein-Barr virus can cause lymphoma. Hepatitis C virus has been implicated in the causation of liver cancer. HPV is well known to cause cervical cancer in women. So in this field of linking cancer with gut microbiota, we are wondering whether some bacteria like Helicobacter pylori may be there to cause colorectal cancer or other kind of cancer. So bacteria or bacterial metabolites are responsible for causing some percentage of cancer in human. So H. pylori, like when we are trying to link bacteria with cancer causation, so this field is quite important, and you can note this when Australian scientist Barry Marshall identified one bacterium, Helicobacter pylori, he got Nobel Prize. So after finding this bacterium, so we are still searching some other bacteria which can cause colorectal cancer or other kind of cancer like pancreatic cancer. So in this field, we are trying to implicate bacteria and bacterial metabolite. And these are the bacteria for colorectal cancer causations like Fusobacterium nucleatum, E. coli, P. plus strain, Enterococcus, S. bovis, Bacteroid fragilis, H. pylori. So all these bacteria have been implicated. Though we don't have sufficient evidence or we don't have concrete proof to prove these bacteria are really causative agents, but like they are secreting some metabolite like colibactin. So colibactin may be responsible for causing colorectal cancer. So here, like we have a lot of bacteria like E. coli or Fusobacterium or Bacteroid fragilis. They secrete some harmful metabolites like trimethylamine, colibactin. And these metabolites can interact with the epithelial cells. They can damage DNA, or they can interact with some signaling pathways. They can make epithelial cell to secrete some cytokines which are inflammatory in nature, and either single event or all these events in a combined way, they can cause inflammation, and finally colorectal cancer, fully blown cancer can be seen. So far we implicated bacterial metabolite bacteria in the causation of colorectal cancer, but there is a field which is upcoming, which is connecting microbiome with the cancer causation. So microbiome is the collective study of fungi which are inhabiting human gut. So Aspergillus, Penicillium, Fusarium species, they secrete mycotoxins like alpha toxins, and they interact with the epithelial cell. They can damage DNA. They can activate some signaling pathway which are implicated in the cancer causations. So these two fields, microbiome and microbiome, these two fields have been implicated in the causation of colorectal cancer, and not only colorectal cancer, but other cancer like pancreatic cancer or gastric cancer. These bacteria and bacterial metabolites have been implicated recently. And so far, we have evidence for colibactin. Colibactin is a large metabolite which is secreted by E. coli and Klebsiella, which are having PKS island in their genome. And these colibactin, lot of papers are out which prove colibactin can damage DNA, and colibactin is responsible for colorectal cancer. So besides colibactin, we don't have other metabolites with concrete evidence. Okay, there are many metabolites, there are many bacteria which have been implicated, but solid evidences we have for colibactins only for the causation of colorectal cancer. So far we discussed about negative roles of metabolite in the causation of cancer, but some microbial metabolites may have positive sides. They may be implicated in the anti-cancer potentials. So in 2021 landmark paper came, it is published in Science, and this paper this paper showed inosine, which is microbial metabolite, can enhance the efficacy of immune checkpoint blockade therapy. So after this, then many paper came to prove metabolites like short chain fatty acid or other metabolites can be implicated for the treatment of cancer. So like short chain fatty acid, there are many papers which discuss about the anti-cancer potential of butyrate or other kind of short chain fatty acid like acetate, valate. Okay, so these metabolites, short chain fatty acid metabolites can inhibit cancer cell proliferation. They can induce apoptosis, or they have different mechanism to kill the cancer. And these are the possible mechanisms through which bacteria can target tumors. These bacteria or metabolites can do autophagy or apoptosis, or they can make cytotoxic T cell to secrete perforins or granzymes. They can interact with the T regulatory cells. So there are so many mechanisms through which these metabolites or bacteria can act against cancer cell. So in my lab, um, I'm screening different metabolites, microbial metabolites for their anti-cancer effect. And I studied 4-ethyl phenyl sulfate, p-cresol sulfate, indoxyl sulfate for their anti-cancer activity. So 4-ethyl phenyl sulfate is a common metabolite now. And from our diet, some gut microbiome can convert dietary component into 4-ethyl phenol. And 4-ethyl phenol goes to liver, and liver can convert 4-ethyl phenol to EPS, 4-EPS, 4-ethyl phenyl sulfate. And then this metabolite comes again to blood for its physiological functions. So we exposed cancer cell, SCT116, and normal cells, and we found like we took different time point, 24 hour, 48 hours, 72 hours, and we found like cancer cell health is compromised at concentrations of 5 millimolar or 10 millimolar. But when we compared these results with normal cell line, we also did MTT assay on normal cell line, CCD841, which is normal colonic cell line. And here we can compare damage to normal colonic cells was not much. So ethyl phenyl sulfate selectively killed a cancer cell, but it is not putting damage on normal colonic cells. Then we did different viability assays like ATP count assay, and we saw the same pattern. We saw like at concentrations of 5 millimolar or 10 millimolar, cells are compromised. We did then cellular imaging. We saw cellular morphology is distorted at 5 millimolar or 10 millimolar concentrations. Then colonogenic assays we performed, and we saw like at concentrations beyond 2.5 millimolar, like colonogenic ability of the cells were compromised. We checked apoptosis, we checked like Roche generations, and we saw like beyond 5 millimolar or 2.5 millimolar, we have compromisations on cell health, cancer cell health. And we also checked through Western blot Bex and BCL2 ratio, and we saw beyond 2.5 millimolar. This ratio was increasing, which is suggesting like apoptosis is happening. We also studied cell cycle arrest, and like GTM cell cycle arrest was seen. Then we checked p-cresol sulfate product effect of p-cresol sulfate on cancer cells. And here we saw same pattern. Like we used SCT116 cell line and normal colonic cell line, and we saw like at 5 millimolar or 10 millimolar concentrations, all these metabolite EPS or pieces, they are damaging cancer cells, but they are not damaging normal colonic cell. So this effect is quite selective, and like cellular imaging and other assays we studied, we saw same pattern when we did ATP count assay or when we did other LDH assay, which is about membrane integrity. And colonogenic assays we also performed for PCS, and we saw like at 5 millimolar or 10 millimolar concentrations, cells are compromised. We did cell cycle analysis and then apoptosis and ROS assays we performed for PCS, and we saw similar result as we saw in the case of EPS. Then third metabolite I selected, that is indoxyl sulfate. Indoxyl sulfate, if we have dietary tryptophan, so some bacteria can convert dietary tryptophan into indole, and these indoles can go to blood and from blood to liver. And on liver, indoxyl can be converted to indoxyl sulfate through the action of sulfotransferase. And then again, indoxyl sulfate comes to blood for its physiological functions. So why we checked indoxyl sulfate for its anti-cancer activity? Because many drugs which are in clinical practice are actually indole derivatives. Drugs like nintedanib or sunitinib or vincristine, vinblastine, all these drugs are indole derivatives. Indoxyl sulfate was also indole derivative, but it was made by bacteria. It is gut microbiota derived metabolite, and there was no paper or no evidence for its anti-cancer activity. So we thought we should check its anti-cancer activity. This was the premise. So here we did the same thing, but in this case we took two cell lines, SCT116 and ST29, and we saw like beyond 2.5 millimolar concentration. So cell health was compromised. Then we did ATP count assay, and we saw same result at 5 millimolar or 10 millimolar concentration. Cell health was compromised. We did LDH assays, we saw same result, and then we did apoptosis, and we did apoptosis, and we saw like beyond 1.25 millimolar concentration, apoptosis is increasing as we are increasing, or as we are increasing the concentration of indoxyl sulfate. Then we did all these assays as we did in the previous with previous metabolites. Then we extended our study to animals, and we saw whether indoxyl sulfate is safe to use. If we consider indoxyl sulfate for further application to use it as a drug, so we gave indoxyl sulfate to animals, and we saw like indoxyl sulfate is not damaging like intestine, or like there was no difference in colon length or spleen length. So indoxyl sulfate is quite safe, and we gave indoxyl sulfate at high concentrations like 50 millimolar intrarectally and intraperitoneally. We did the histology study, and we see when we compared with the vehicle control. So colon tissue and rectum tissue, we didn't see inflammation or any damage to the tissue after exposing of indoxyl sulfate. We also checked inflammatory cytokines after giving indoxyl sulfate, and we saw like in cytokine level like IL-17 or IL-1 beta or TNF alpha, these kind of cytokines we checked, and we saw like the difference is not significant with the control. So there was no inflammation after giving indoxyl sulfate to animals. So this is the conclusion. Our results showed indoxyl sulfate is responsible, not indoxyl sulfate, but other metabolites. They are responsible for inducing apoptosis, cell cycle arrest, loss of membrane integrity. And all these metabolites are killing cancer cell in a very selective way. So they are killing cancer cells, but they are not harming normal colonic cell, or they are not harming colon part of animal.
Time. So I think you have talked about the different metabolites which have anti-cancer role. But in the gut homeostasis, when it is there, do you think it is the one metabolite, or it is the imbalance of the metabolites which causes cancer versus which are anti-cancers, which are, leads to the cancer formation? So I believe as a clinician that it is mostly the imbalance of the anti-cancer versus cancer-causing metabolites. What do you think about that?
Okay. So some metabolites like colibactin. So it is not about balance, but if colibactin is secreting, if some bacteria like PKS plus E. coli or PKS plus Klebsiella, if these bacteria are present and they are secreting colibactin, whether colibactin is secreted in less amount or large amount, if colibactin is there, then it is sure these metabolites will damage DNA, and this DNA damage or inflammation can cause colorectal cancer in long term.
Okay, the talk is open for discussion. Yes, sir.
So in continuation of what you said, see, you mentioned both the bacteria which are pro-cancer, like the metabolites are promote cancer, versus those metabolites you later showed in your study which act as anti-cancer. Now, these two populations in the gut would again depend upon the diet. So what are the dietary linkages to the different populations will actually ultimately translate into effective interventions. While the metabolite in the therapeutic approach is a different one, the linkage with diet is also equally important if you want to prevent this.
Okay. So diet is one factor, but infection of these bacteria is other factor. Like Salmonella typhi, if we get infection of Salmonella typhi or H. pylori, okay. So some bacterial or like PKS plus E. coli strain or Klebsiella. So diet can modulate the bacterial population, but infection is also one factor. We can contract these bacteria from other person or from environment, and if we are infected by these bacteria, then they can secrete these genotoxic metabolite or genotoxins.
I will take what Dr. Malhotra was saying. We'll take it further. Since yesterday we were looking at different papers which were presented about the microbiome population in the colon cancers, and there were distinct signatures of different types. So colibactin can cause cell damage, DNA damage in cells, but it can also eliminate the other bacterial community. It is a kind of a bacteriocene. So it might eliminate some of the other beneficial organisms and ultimately change the environment. So it will be interesting to see the microbiome population in the animals or subjects where the colibactin, enhanced colibactin is being produced. So the disturbance of the communities or environment in colorectal cancer could be equally important.
You are right, sir. So there are many comparison reports between healthy people and cancer patient. Their microbiota is different. You are very right. Like these metabolites can alter the microbial population. You are right.
I have an extension of Dr. Shashikaran's question. So basically, indoles, phenols, and cresols are produced as a result of bacterial metabolism, nitrogenous food. So it's proteins which are not absorbed in the small intestine, which escape into the colon, which lead to these. And sulfation happens, sulfation of these indoles, phenols, and cresols happens both in colonic epithelium and in the hepatocyte. It's actually a mechanism by which the body detoxifies this and excretes it either in bile or in the urine. So I think what you have shown is probably that indole sulfates can have an anti-cancer effect. You're trying to use that as a drug. But in nature, what happens might be that these indoles phenols and cresols may actually be carcinogenic in a different way. It's possible.
Yeah, actually, these indoxyl sulfate and paracrysyl sulfate, ethyl phenyl sulfate, all these are uremic toxins. Okay, so these metabolites are quite negative in their nature, but they also have positive sides. Okay, we studied their anti-cancer activity, so they have dual functions. We don't know like uh, what is the mechanism, but we found anti-cancer activity of these metabolites.
My question is that I'm a microbiologist and we know E. coli and Klebsiella are normal commensals of colon, right? So as you mentioned, there is a island of genes which is responsible for colibacter production. So what percentage of these bacteria carry these genes? Because they are normally present in the intestine.
Okay. So not all E. coli have PKS islands in their genomes, but some specific strain of E. coli. So they are generally not present in healthy people. They are present in people like who are having colorectal cancer. So in deceased person, they can be found, but I don't think they are present in normal. So when we compare metagenomic analysis, healthy versus cancer patients, so they are not present in healthy
So
people.
can we identify their source in the environment, how these people are acquiring? Can we
Okay.
work on that?
Okay, I don't know that how like E. coli
I
PKS
think
plus.
further discussions during tea time or the lunch time. Please to stick to the time. Thank you so much, Dr. Anil Kumar, for your interesting presentation.